Myself, Shaik Abdul Karimulla, working as an assistant professor in YP CRC in Department of Pharmaceutical Chemistry. So, as a part of NMedia presentation, I'm going to explain about pharmacophore modeling and dogging techniques. So, these are the specific learning outcomes. After the completion of this presentation, student will able to say about introduction to CAD, different types of computational approaches of drug design, and what is pharmacophore modeling, and the basics of docking and different docking techniques. So these are the content that I will discuss in this presentation. The first thing is introduction to the CAD and what are the types of in CAD and pharmacophore modeling, and finally we'll discuss about docking techniques. So this is about introduction to CAD. So exactly CAD means computer aided drug design, which refers to the use of computational tools and techniques to design and optimize drug candidates. It plays a crucial role in modern drug discovery by saving time and reducing costs. So before the developing of computer aided drug design, the scientists had been depending upon the conventional way of drug design, in which the scientist will go for natural extraction, and with the knowledge of structure activity relationship, they have been modifying the existing drugs. That's what we have been doing till now, before the discovery of computer aided drug design. But after the developing of computer aided drug design, the scientist work has been made easy. In which way means, so we'll use computational tools to design the drugs with the knowledge of the back history. Like we have been having a lot of chemical libraries as an information. Based upon that knowledge, we'll go for drug design. And in the CAD, three key features are there. The first one is virtual screening of compounds, and the second one is prediction of interaction between drugs and biological targets, and third one is optimization of drug chemicals. In the virtual screening of compounds means, we'll screen the lot of compounds present in the chemical library. After the screening, we will find out what are the chemical compounds having affinity towards a particular target. Based upon that information, we will go for hit identification. After that, lead identification. Later, we will go for lead optimization, and finally we will be having a final compound which will be having the ultimate affinity towards a particular target. So after that prediction of interaction between drugs and biological targets. After the developing of drug compound, we will go for prediction of interaction between drugs and biological targets by helping of docking tools. In docking, what we actually do means, we will go for checking of affinity between drug and biological target, and after that optimization of drug compound based upon the information that we obtain after docking. We will go for optimization, in which way means we will keep on adding or deleting some chemical elements from the structure, so that it will be having more better affinity, and it will be having more pharmacokinetic properties. So why CAD only? So as I said before, in conventional way of drug design, it had been taking a lot of time. For example, whatever it may be the drug compound, in the ancient days, it took at least 2 decades to come into the market. But with the help of CAD, the development has been made easy. So it speeds up the drug discovery process, and also reduces the dependency on trial and error methods, and also enables the exploration of vast chemical space. So in computer aided drug discovery or drug design, we'll approach two methods. One is structure based drug design or receptor based drug design, and the second one is ligand based drug design. In structure based drug design, we utilize the 3D structure of the biological target to design new ligands, and pharmacophore modeling aids in de novo drug design and target identification. This is about structure based drug design in a brief way. And coming to the ligand based drug design, it involves the relies on knowledge of other molecules that bind to the biological target. So pharmacophore modeling includes virtual screening and lead optimization. In structure based drug design, we depends upon the structure of target molecule. Based upon that, we will go for drug design. But in the ligand based drug design, we are depending upon the already existed ligand molecule, so that we will go for developing of new entity in such a way it will be having more affinity and more efficacy. So this is a simple flowchart about computer-aided drug design, which go for target identification in two ways. One is structure-based and ligand-based. In structure-based, we'll go for after preparation of structure, we'll go for high throughput docking. In ligand-based, after studying structure-activity relationship, we'll go for pharmacophore mapping. Coming down to these both involves the hit identification, after that hit to lead optimization and finally in vitro verification. So this is the pharmacophore modeling. Before going into in detail, what is pharmacophore? According to IUPAC, a pharmacophore is a group of features in a molecule like shape, sizes or electric charges that interact with a biological target like a protein or enzyme or trigger or block a biological response. Pharmacophore means it's like a structure which need to be have properties like based upon the pharmacophore shape. We will go for design so that the developed molecule will be having desirable qualities. It's essentially the blueprint of what a molecule needs to fit and work with a specific biological target. So the purpose of pharmacophore modeling means it explains how structurally diverse ligands can bind to a common receptor site and assist in identifying novel ligands through de novo design or virtual screening that will bind to the same receptor. So the workflow of pharmacophore modeling involves that first we will go for target identification and validation. Say for example, if any new disease arrived based after conducting so many research, we'll be having knowledge about pathophysiology of that particular disease and root cause. Root cause may be any enzyme or a protein or a receptor. This is identified as a target. After that, we'll go for ligand selection and through collecting a set of known active molecules from a chemical library. Or if it is not there in chemical library, we will go for structure design. And after that, we will ensure that structural drivers to create a robust pharmacophore modeling. And after target identification and validation and ligand selection, we will move on to the pharmacophore model generation, in which we will identify common chemical features like hydrogen bond donors or hydrogen bond acceptors or hydrophobic regions, etc. among active ligands. Based upon that information, we will go for generating a 3D pharmacophore model representing these features. And later we will go for pharmacophore model validation. We will validate the pharmacophore model that we generate before. In this, we will test the model against known active and inactive molecules, and after that we will ensure it accurately distinguish between active and inactive compounds. We will test this pharmacophore model whether it is distinguishing a difference between active and inactive compounds or not. And later we will go for virtual screening, in which we will use the validated pharmacophore model to screen large chemical libraries, and later we will go for identification of compounds that match the pharmacophore features. And after virtual screening, we will go for hit identification. Hit identification means the compounds which are having at least optimum affinity or efficacy towards the target molecule. Here we will filter potential hits from the virtual screening results based on the pharmacophore match and prioritize hits for further analysis. And after hit identification, and we will go for molecular docking and scoring. We will go for molecular docking between hits and a particular target molecule, in which we will perform docking studies to assess the binding orientation and interaction of hits with the target. After that, we will rank the compounds based upon the binding affinity in a descending order. After that, lead optimization. Based upon the docking score, we will go for lead optimization, in which we will modify the structure of identified hits to improve binding affinity, selectivity and drug-like properties. And here we use the pharmacophore model as a reference for the optimization. After that, after lead optimization, we will move on to biological testing. We will test optimized compounds in vitro and in vivo to evaluate biological activity and therapeutic potential. Later, we will go for final selection of drug candidates. After biological target, we will go for final selection of drug targets, in which we select the most promising compounds for preclinical and clinical development. This workflow integrates computational and experimental steps, accelerating drug discovery. So this is a virtual screening in brief, where we input chemical database and later we will go for development of pharmacophore model and screen compounds against the pharmacophore. After that, we will filter hits based on fit score, and later we will perform docking studies between a drug molecule and a target. After that, we will select potential drug candidates And this is the basic difference between ligand-based pharmacophore modeling and receptor-based pharmacophore modeling. In ligand-based pharmacophore modeling, we go for in ligand-based pharmacophore modeling, we require a set of active compounds with known activity, whereas in receptor-based requires the 3D structure of the receptor protein ligand complex. Source of information in ligand-based pharmacophore modeling, it relies on common structural and functional features of the ligands, whereas in receptor-based pharmacophore modeling, it relies on uses the binding site architecture of the receptor. After that, techniques include QSAR molecule alignment and common feature analysis, whereas in receptor-based pharmacophore modeling, we go for techniques like docking, molecular dynamics and active site mapping. So both have their respective advantages and disadvantages. So we will move on to the technique based upon our desirability. So this is the docking and molecular docking, the most important part in computer aided drug design. So what is docking? Molecular docking is a method used to predict how two molecules like a drug and protein will fit together. So it helps in scientists understand how well a drug might bind to its target, such as a disease causing protein, to work effectively. Think of it like trying to fit a key into a lock. Molecular docking simulate how the key fits into the lock to see if it will work properly or not. So these are the key steps that involved in any molecular docking. The first one is preparation of structure of a protein and binding site identification, docking simulation, scoring and analysis, and finally validation. Preparation of structure which involves obtain the 3D structure of the target protein, often from databases like the protein data bank, ensure the structure is complete and optimized for docking, which means, say for example, if we are having a new disease developing, after research we will be knowing about the pathophysiology and we will know about the root cause, which is protein or enzyme or a receptor. So if the receptor has been reported already, we will be having information of that particular receptor in PDB database or protein data bank. There we will be having 3D structure. From there we will obtain the protein and we will prepare the protein according to our desirability. If we want to go for docking for a single chain which is involving in that particular protein, we will only select that single protein which is having active sites. After that, ligand preparation. So we will search the chemical library that already existed. Otherwise, if you want to develop a new compound, we will design a scheme and develop a new drug. So we will obtain or design the 3D structure of the ligand, example a potential drug molecule, and later we will optimize its geometry and assign the appropriate charges. So after that, binding site identification. So protein preparation and ligand preparation are the two key points. After that, binding site identification is the another step involving the docking, where we will determine the potential binding site on the protein where the ligand may interact. This can be done through computational tools that predict the binding pockets. And after that, docking simulation. So by using any software tool, we will go for docking simulation where we simulate the actual biological interaction between protein and ligand. The software explores various orientations and conformations of the ligand within the binding site to predict the most favorable binding mode. So scoring and analysis. After the completion of the docking software itself gives the score and we will go for analysis, in which we will evaluate the predicted binding modes using scoring functions that estimate the strength and stability of the interaction. After that, we will analyze the result to identify the most promising ligand protein complexes. After the scoring and analysis, we will move for validation. In here we will go for validation of the docking result through experimental methods or further computational analysis to confirm the predicted interactions. So these are the key stages in docking. First we will target our protein selection and preparation. After that, ligand selection and preparation. In target selection, the 3D structure of target or protein can be downloaded from PDB database. In the ligand selection, ligand can be selected from PubChem or ChemSketch or ChemDraw. So in target selection, the receptor must be biologically active and stable, whereas in ligand selection, the ligand should be docked onto the receptor and the interaction should be checked. In target selection and preparation, target or receptor may have many active sites. Interested one should be selected and included. Whereas in ligand selection and preparation, the scoring function generates the more depending score depending upon which the best fit ligand is selected. So these are the normally using general docking tools, where we can see autodock, Vena, dock, molecular operating environment, simply MOE, and glide or Schrodinger, flex, pyrex, pymol, open babel, and these are some newly developed docking tools which working upon the AI generation. So these are deep dock, Gnina, AlphaFold, Multimer. So for example, the breast cancer is developed by the mutation of BRCA1 and BRCA2 proteins. These are tumor suppressor genes involved in the DNA repair. If any mutation in these genes significantly increases the risk of breast and ovarian cancers. After that, so the root cause for breast cancer in mutation in BRCA1 and BRCA2. After that, it can be chosen as a target for experimental molecule or ligand or test molecule. After that, we will go for ligand selection. Here, for example, I have chosen a doxorubicin. The above selected target can be docked against an experimental ligand drawn from PubChem or ChemDraw. So this is the first step, which is target preparation. I prepare this target by using a tool called Discovery Studio. Actually it's an visualizer. So I downloaded the structure from PDB database. After that, this is the Pyrex free docking software, where I uploaded the prepared protein into this one, and this is the grid arrangement. Before the grid arrangements, I incorporated both ligand and protein into this grid. After that, we will move on to docking. So this is the docking source. Even though I have included only one target molecule, it has given 9 conformers. Means if the compound is having so many bonds right here, so depending upon the flexibility, it will bind into this in different manners. So depending upon the mode of binding, it will give different scores. So out of above 9 conformers, the first one is having highest docking score, which means it has the highest binding affinity among all the conformers. So this is the visualizing ligand conformer in Discovery Studio. So this is the 3D interaction by using Discovery Studio, and it is a 2D interaction. So in 2D interaction, we will be knowing about what are the bonds that are present between target and ligand molecule. So here I have shown different bonds in different colors. So after completing docking and analyzing the result, the following steps are typically undertaken, like validation of docking result, molecular dynamic simulation, binding free energy calculations, experimental validation, structure based optimization, virtual screening, publication and reporting of document, and further computational screening. So in a summary, a pharmacophore represents the essential chemical features required for a molecule to interact with a specific target, and it will be used to identify and design new compounds with the desired biological activity. And it has applications like virtual screening, lead optimization, and understanding structure activity relationship. Coming to the docking, it is a technique simulates the interaction between a ligand and a target molecule, and it will predict the binding modes, orientations, and binding affinities. And it has some applications like drug discovery, binding site prediction, and structure based virtual screening. So these are the reference from where I have drawn this content, and thank you.